Hi, everyone. I'm Dr. Sharyn Rossi, the Senior Director of Neuroscience Programs at BrightFocus Foundation. BrightFocus has been around for over 50 years, funding innovative research in Alzheimer's disease, macular degeneration, and glaucoma, three age-related neurodegenerative diseases that we're very interested in finding treatments and cures. I've been here for about 5 years. I came from the National Institute on Aging and have a diverse background in many different things, neurodegeneration and repair. But I'm most excited today to have a conversation with my friend and colleague, Dr. Keith Hengen from Washington University in St. Louis. And Dr. Hengen is a grantee of our Alzheimer's disease research program, and he's here today to talk a little bit about the really exciting work that he's doing. Keith, welcome. Would you like to introduce yourself?
Yeah, I'm Keith. I'm in the Department of Biology at WashU, and I would say I'm a systems and computational neuroscientist. And I came from a background in hibernating ground squirrels, and then I studied pregnancy, and then I moved to kind of the fundamental mechanisms that allow brain computation to be stable, which eventually led me to BrightFocus and trying to tackle the problems related to neurodegeneration and Alzheimer's.
Yeah, that's really interesting. So you're hitting on a really interesting point that Bright Focus is really excited to bring people from outside the Alzheimer's disease research space and utilize their knowledge in some of these other areas to really bring their expertise to the disease space to help us better navigate such a complex disease. So as Keith said, he's a computational neurobiologist. And in lay terms, what does that mean, Keith?
In lay terms, computational neurobiology. Um, Seems like an oxymoron. It basically means trying to understand the mathematical principles of how some aspect of the brain works. And in this case, sort of a process of subtraction. So if you can compare a diseased brain from the way information flows through it to a healthy brain with the way information flows through it, you can start to define what's actually going wrong in that brain.
So information flowing through a brain, it sounds a little bit, can you maybe compare a brain to a computer?
What a setup. Yeah, so I think that I take the perspective that a disease is not a protein or a molecule, right? So a molecule and a protein can certainly cause a disease, but there's a difference between cause and effect. Two central suspects in driving Alzheimer's are tau and A beta, and in my mind, they are not intrinsically the problem. The problem is that they cause some deficit in how a brain computes, how a brain works, whether you're computing, trying to remember something like where you parked your car, what you ate for breakfast, or access to your sort of full lexicon of your vocabulary. That's the actual problem. And as a field, the issue in my eyes is that we have no idea what distinguishes that kind of mathematical aspect of a brain with Alzheimer's from one that is healthy. It's not simply that the neurons aren't firing. There's some fundamental difference in how they compute and how they transmit information that is truly the basis of the symptoms of any disease, and we don't know what those are. And so that's the angle that we come in at. Yeah.
Yeah, and what an interesting angle that is, unlike most people in this space, I think. And so, have you identified maybe any one underlying principle or mathematical phenomena that drives this kind of brain change?
Yeah, so I think from first principles you can say it has to be a failure of homeostatic plasticity. And so that's
Now,
a
explain
jargony
that a
term.
little bit more.
No, I know, I know, I know. So that's sort of a jargony term for basically the thermostat, right? That there's some set point that is necessary for normal functioning, sort of the way your house might be set to say 72 degrees. And it doesn't matter if you open the windows in February because you'll sense that and you'll kick on the furnace and you'll stabilize temperature. The brain is there's many mechanisms that try to ensure stability. And so sort of by definition, you can kill off a bunch of neurons or have a bunch of problems. And as long as that thermostat can adjust for it, it doesn't matter. I mean, you probably lose many neurons every day. And as long as you can compensate for it, it doesn't matter. So we kind of approach the problem by saying, well, some aspect of homeostasis, some thermostat either is broken or can no longer counterbalance that cold air coming in. And the place where we really see the problem explode is at the level of information flowing across many networks. It's not at the single neuron level. It's the way hundreds of thousands to millions of neurons actually pass information around. So the the specific mathematical principle here relates to the fact that if you imagine some big network of neurons like in your brain, and you're somehow tuning how readily they transmit information, it's not like for every little twist of the knob, the brain gets better and better and better and better. It's basically you could twist it, you twist it, you twist it, nothing happens, nothing happens, nothing happens, and you cross a line and it explodes into a seizure. And so this is this edge of chaos phenomenon where the system goes from just damped and inert to just chaotic like that. But it turns out that the very last steps before that chaotic regime, it's like a blossoming. Like all of a sudden, any information processing suddenly emerge and are kind of optimized right there. And so in a healthy animal, like that is very clearly the point that sort of development and evolution and homeostasis are continually trying to restabilize. That seems to be the endpoint of sleep in the brain. And interestingly, that's what starts to fall apart very, very, very early in Alzheimer's and other neurodegenerative diseases. Like long before symptoms, we begin to see that the brain starts to drift away, 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 away from that point. And as you get further and further and further away from it, all of a sudden the symptoms start to come up. So it's both a good early warning sign, definitely in animal models, and now we can show this also in people, but it also provides sort of a measure of the depth of the disease as well.
So you said early warning sign, and I think, for the Alzheimer's disease field, we talk a lot about early diagnosis because, as you kind of alluded to, a lot of the changes that are occurring are very early on in life. I mean, we can detect protein changes at midlife that are cardiovascular type proteins that can confer risk for Alzheimer's disease later on. And so can you speak a little bit to that, why early diagnosis is so important, and how early can you detect using your method, and what types of degenerative processes can you even detect using your method?
Sure. First, I'm going to just start on the why is early detection important. And again, I am not a clinician. Spent a lot of time talking to clinician scientists, and it seems that there's a general agreement that we probably have effective drugs, effective interventions right now. But by the time we know somebody actually has a disease, they might have lost already 70 to 80% of a given network. So, for example, in ALS, by the time you have a respiratory phenotype, problems breathing, you've already lost at least 75% of the phrenic motor neurons. Those are the neurons that actually drive your diaphragm. So, effectively, once you know you have a problem, there's nothing really left to save. So, early diagnosis then would let you into the system before it's failing, right? So that's sort of the sort of really just baseline obvious importance of early detection. And at least in animal models, we have an extremely high resolution on tracking what neurons are doing. We can see that the system is sort of departing from the normal lane in the equivalent of like a 15 or 20-year-old human, right? But again, these are animal models of the overexpression of certain genes that, for people who have familial Alzheimer's, we take that gene, we put it in a mouse, and we can see this departure very, very early in life. I obviously don't want to kind of conflate that with human studies.
Well, so this is by directly recording brain activity in those animals. So how would you do it in humans?
So again, through our clinician collaborators, we've gotten access, at least to right now, to some pilot data sets of people who are cognitively normal in middle age, who have no familial history of Alzheimer's, then cognitively normal people in middle age who do have a history, so they're predisposed to it, and then we have people with mild cognitive impairments. That's kind of the prodromal early stages of Alzheimer's. And what we see is that that first group of people who are probably never going to get the disease are very, very close to criticality. The group of people who cognitively don't have any symptoms yet, right? At least any that they could put their finger on, they're a bit further away. Statistically, we can show that they're further away. And then the people who actually have the beginning stages of Alzheimer's, they're beginning to manifest some symptoms, they're the furthest away. And presumably, not that you usually scan people in the end stages of a disease, because that would kind of be inhumane, but that kind of just takeoff where you just start to really fall apart away from this critical point would only get deeper and deeper and deeper as you go further into the disease. So the principles hold. This, I guess what I'm trying to get at is that I can't directly nail down the time course of comparing this mouse model to people, but as far as we can see in people, we should be able to differentiate folks before there are symptoms.
So that's interesting. So you're talking a little bit about a threshold. So I mean, in a normal life experience, what do you think are the variety of insults that could be pushing one's homeostasis past that threshold? Is it a combination of things? Is it all the exposures that we're exposed to in life and that mixed with genetics?
Yeah, I think you've got your finger on it that it's going to be an incredibly complex mixture of things. And Um, I take this isn't a contrarian view, but it's certainly not the standard perspective. I think that neural activity itself probably determines something of the fate of the brain. And we take this for granted when it comes to short-term changes. Like that's what plasticity is, like learning and remembering somebody's name or a phone number or recalling where you left your keys requires synaptic changes. And it is the timing of activity in the brain that actually does that. So the way we're looking at this is it's just a longer time scale. It's like imagine driving your car in the wrong gear for thousands of miles. Like it might work, you might be able to get where you're going, but over time you're going to start to get sort of shards of metal in your oil. You're going to start to destroy the engine. And so um, analogously, if the brain is sort of at the wrong set point or it can't get back to the right setting on that thermostat, I think that there's sort of a cumulative damage that happens over time. And that is really well poised to explain why when you have twins and one of them gets Alzheimer's very early and very, very severely, and the other doesn't or gets it much more mildly, much later in life, genetically they're basically the same. So there's got to be something about experience, the patterns of activity in the brain that actually can deterministically send one of these people into a much worse fate than the other.
So do you think that the patterns are going to be that unique that they're kind of personalized signatures, or do you think that you would have disease specific signatures? Like, does one disease look the same as far as neural activity goes?
Oh, okay. So that's sort of two different layers of questions. So that distance from criticality is best thought of more like blood pressure, right? That blood pressure isn't specific to this heart disease versus that, but it is an extremely valuable kind of prospective diagnostic of upcoming catastrophe. And so for us, to simply say that this brain is far away from criticality, I can't tell you whether it's Lewy body dementia, Alzheimer's, or Parkinson's. But if you were to say, oh, this is a 50-year-old person who is a ultramarathoner, and we see problems around criticality in motor cortex, I'd say that's probably ALS, right? So the anatomical pattern and the demographic data help you really hone in on what that might really be indicating more specifically. Um, but in terms of the patterns, so the personalized patterns, what you do fundamentally determines the activity in your brain. So if you tend to, we think sleep restores criticality. That's kind of the why all animals need to sleep is that experience pulls you away from criticality, sleep brings you back. And that would sort of tightly and neatly explain why shift workers and people with really bad sleep habits, whether that's voluntary or not, get worse neurodegenerative disease, right? It's causal. This is not just correlational anymore. I think it's sort of very well established. And so they're basically keeping the system from getting back to its set point. They're driving the car in the wrong gear. And so that would be a way, for example, that two twins who are genetically identical could end up in very, very different places from a neural dynamics perspective.
Wow, that's really interesting. I mean, I've thought a lot about shift work in that context, but maybe people need to rethink their shift work. So, I mean, it's clear that you're so incredibly passionate about your work. What do you have any driver, a personal driver for especially moving into the Alzheimer's disease field, or why are you so interested in the brain?
Well, okay, so it's almost like you know me. So I think there's two reasons that I'm drawn to work in Alzheimer's, and as you mentioned, like one is very personal, and one of them is I mean, it's personal as much in as much as like I have an academic passion for this central question. So if we start with that, if you really want to figure out the mathematical principles of like why brains, generally speaking, work, one of the best ways to do that is to take a brain that doesn't work, especially one that is progressively failing, and say, well, what of it, what aspect of that is different than the one that does work? Right? Because again, for example, in Alzheimer's, it's not that the neurons just stop firing. It's not that they all just start firing in a tango tempo, right? So you can say, well, what is it? What is it that somehow differentiates these two things? And so you can use the disease state to actually illuminate what it is that is necessary for a brain to work well under normal conditions. And that's a very, very powerful possibility in my mind. But on the personal side of things, my dad died of early onset Alzheimer's in 2016. And that was like, I don't know, beyond description. It was the hardest thing I've ever been through, like hands down. And I felt so just useless because I was, recent PhD in neuroscience, I'm studying the brain and I'm watching this man just slowly evaporate. And it felt like everything I had done was just for folly. Like I could, what I mean? I could read as many papers as I wanted, and it just felt pointless. Like we were just spinning in circles for some hall of mirrors effect. And I thought to myself, like, let's put it this way. Before that point, I kind of saw neurodegeneration as like not even a neuroscience question. It was like a medical question, and it's not really fundamentally about how the brain works. But after having gone through that, I don't care. Like this needs to be solved. And so the fact that I can potentially bring in some of our really, really foundational questions about brains to maybe move the needle on this, I mean, it became kind of just there was no question here. Like there was just full speed ahead. But then it was an issue of how do you get into this field? And that's where BrightFocus is you guys are a godsend.
Wow, thank you all, and thank you for sharing that with us, Keith. And I think on that note, you touched on sleep a little bit, and I'm sure that the audience has probably heard a lot about lifestyle interventions. So we're going to move a little bit in that direction just to end on a hopeful, happy note of what we can do. So again, you touched on sleep. Sleep hasn't been added yet as a risk factor to the Lancet Commission for Alzheimer's disease risk, but we're all waiting for it to be in the next few years. So, I mean, you touched a little bit about the importance of sleep, but maybe we could just end on what people can do today for better brain health.
I think that, and again, I need to restate here, like I am not a clinician, so this is just my perspective from the kind of computational neurobiological perspective and the experiments we do in the lab, right? But I think sleep is so deeply foundational to the brain's ability to kind of function. By that, I mean compute normally and healthily, which is the opposite of Alzheimer's disease, right? That is a degradation of that very process, and sleep is central to that thing. So I think that the idea of taking sleep more seriously is of inevitable benefit in this context, in any context, really, honestly, but even just your own like emotional stability, but particularly with regards to these diseases. And to me, what that means is not just being in bed for a certain number of hours, but actually having clean, consolidated, deep sleep. And so that might mean, for example, not bringing a cell phone upstairs and having a little bit of a better routine and having a blackout shade and a white noise machine and like let yourself sleep to the point where you wake up. It's crazy. Like the first few nights I try to do that, I have small children. I'll sleep for like 14 hours. Not that I could ever actually do that. But then you get
Nice
to the point
try.
where you're refilling the bucket, like you're no longer in deficit, and all of a sudden, after 7.5, 8.4 hour, whatever it is, you'll just start to wake up naturally. And if you could let your brain do that each night without interruption, without kind of getting up 5 or 6 times in the middle of the night, when we can actually causally push animals to have deeper sleep, we can massively delay the timing of the onset of these diseases and mitigate the impact of them. Right. So I mean, it is a potent lever in determining this stuff.
Wow, well, that's definitely a hopeful ending that somehow we can modulate sleep early on in life as an intervention to this deviation from criticality. Is that kind of the overarching idea?
Yeah, yeah. I mean, it definitely is. I think that the details need to be worked out on like which diseases and which genes and what the right protocols are for this, and how do you translate a manipulation of an animal's sleep to human sleep? Those all need to be worked out. But I think that the core principle is conserved.
Yeah. Okay. Well, thank you so much for joining us today, Keith. We are very happy to have you on board as a grantee and also here sharing your expertise with the general public. All right. We'll see you later.